right. Good evening, everybody, and welcome to our second PharmTalk lecture series. My name is Jeff Javier, and I'm the Director of Communications for the University of Arizona R. Ken Coit College of Pharmacy. On behalf of our Dean, Rick Schnellman, I just want to thank you for joining us this evening. I'll begin tonight's event by respectfully acknowledging the University of Arizona is on the land and territories of indigenous peoples. Today, Arizona is home to 22 federally recognized tribes, with Tucson being home to the O'odham and the Yaqui. Committed to diversity and inclusion, the university strives to build sustainable relationships with sovereign Native nations and indigenous communities through education offerings, partnerships, and community service. So the PharmTalk lecture series is an opportunity to learn more about the breadth and depth of the pharmacy field and the impact our college has on addressing healthcare challenges. Whether you are a healthcare professional or simply interested in learning more about the field, I hope you find tonight's series of talks both informative and engaging. Thank you to our speakers, staff members, and the captioners from the Disability Resource Center for making this lecture possible, and to all of you for taking the time to join us. I'll now introduce our first topic. The unprecedented availability of large-scale biological data has paved the way for discovery in precision health. In this talk, titled From Big Data to Precision Medicine, Predicting, Preventing, and Understanding Adverse Drug Reactions, Associate Professor Jason Karnes will discuss the All of Us research program, which curates genomes, electronic health records, and survey data for hundreds of thousands of diverse participants. With a focus on immune-mediated adverse drug reactions, Dr. Karnes will describe how big data can be leveraged to inform our knowledge of genomic influences on drug response and to translate clinical interventions for improvement of patient care. Please join me in welcoming Dr. Karnes to the stage.
Thank you. Thank you so much, Jeff. I really appreciate that introduction, and it's an honor and a pleasure to be here. Thank you for joining us. This is quite a title, but I hope to distill it. I hope to make it a little bit more accessible. And we're going to talk a little bit about one of the major initiatives here at the University of Arizona, which is the All of Us research program. And I'm going to do this in a discussion of big data. All right, so what do we mean by big data? We're talking about essentially the explosion of biological data that we're now able to produce. There's many methodologies that are relatively new that we're just churning out more and more data. And we're doing it at a rate where we don't really know exactly how to use it. And then precision medicine, which I'll explain a little bit, but we're trying to utilize that data in order to personalize treatment. Getting away from sort of the guess and check, trial and error sorts of approaches that are very common in today's medicine. So what do I mean by guess and check? Really what we're talking about is you go to a primary care physician, you have hypertension, they guess which drug is going to be best for you. They may utilize some of your characteristics, your age, your weight, your race and ethnicity even. But in the end, we're making these decisions based on averages. The average person within a population is going to respond to this blood pressure medication. In this particular example, I have statins. So statins are a cholesterol-lowering medication. They prevent heart attack by lowering your cholesterol. And they have something called a number needed to treat of 10. And a number needed to treat is essentially how many people do you need to treat with this medication to prevent one heart attack? And this is 100 individuals up here. So if you treat all 100 of these individuals with a statin, you prevent 10 heart attacks. Those are the individuals designated in yellow. But you'll see this is a 40% risk population, and so 30 of these individuals are going to get a heart attack regardless of whether they got the statin or not. All these other individuals, they're not affected by the statin treatment. They wouldn't get a heart attack or get one prevented. And then we have a small percentage that's also going to be subject to adverse reactions. They might discontinue the drug because of side effects. And that's what I mean by adverse drug reactions, is really side effects of a drug. They can be severe or they can be minor. And this isn't specific to statins, right? We have this number needed to treat for very many of the commonly used medications out there. But my point is, we can do better, right? We can, instead of just giving this drug to 100 different individuals to save 10, which is worthwhile, absolutely, how do we differentiate between who's going to benefit, who's not going to benefit, maybe can get an alternative drug, and maybe isn't subject to the costs and the potential risks of this drug treatment by this on average guess and check style approach in medicine. So precision medicine is where hopefully we're headed. So this is an emerging approach for disease treatment, and it takes into account the variability within your genes, within your biological data. Sometimes we call these biomarkers. This could be anything as simple as a cholesterol level, or it could be a whole genome. It could be your whole genetic makeup. And you can see here in the middle of the slide, this is the standard approach. We do a randomized controlled trial. We say on average, this is the best drug in this population. But we don't necessarily cater to an individual. We don't cater to individual characteristics. We just use the population average to treat the individuals with the drugs that work best on average. But who's to say that drug will work best for you? So precision medicine, what we're trying to do is we're trying to incorporate that genomic information as well as some of the other biological information that we now have access to. Things like the transcriptome or RNA sequencing, the metabolome, which is the small molecules within the body, the microbiome, so an emerging area where we have bacterial sequencing. We can tell all the bacterial species and the fungi and the viruses that are present on all different surfaces of the body, and then the list goes on and on. So the hope is that we use this data to streamline treatment, to effectively establish who can benefit most for these treatments. So here's an example. Here's a concrete example with something called HLA. It's human leukocyte antigen. These are the same genetic markers that are used to match organ transplant recipients, right? So when you say they're a match, that means you're getting a kidney from a family member, you're matched based on your HLA. So there's an immune system involvement here. But HLA can also be used to predict side effects of drugs. One specific one is abacavir. This is an anti-HIV drug, and we can use HLA typing to predict whether or not someone's going to have this severe reaction to abacavir. And now it's standard of care. HLA screening is recommended for anybody who gets this drug. And that's because people with the variant are at 30 times higher risk for the reaction. Interestingly enough, this drug wasn't utilized at all 10 years ago because it had this very serious reaction associated with it, this hypersensitivity reaction. Now it's made a comeback. Now people are utilizing it again. Number one, because HIV resistance is on the rise and old drugs can be very effective. But number two, we've essentially eliminated this side effect from possibly even happening in a patient. And so physicians are now using the drug more and more often. But one thing to note here is how this variant really only occurs in individuals with European ancestry. It's virtually absent in any other, in any non-white group. And there's major differences in how common these alleles are across these different populations across the US. Now keep that in mind. This is a complicated slide, but just to say that we've been doing work in HLA here at the university. At our Ken Coit College of Pharmacy, we are leaders in this line of research where we're using HLA polymorphisms to predict severe adverse drug reactions to different drugs. So, a couple of questions here, because I mentioned this disparity, how this HLA testing is really only effective in white individuals, right? In non-whites, there's a low prevalence of the allele. We don't understand as much about how those alleles can predict adverse drug reactions. And so are the genetic results that we're getting, are they informative for individuals that maybe don't have European ancestry? And similarly, there's a cost associated with this sequencing, with this HLA typing. Is this only going to be available to individuals that have the means, to individuals with premium insurance, to individuals that can essentially afford it? And then one of the things I want to focus on today is, what are we missing out on when we look exclusively in white populations, in individuals with European ancestry? And I'll try to make the case that diversity is going to help us all from that perspective. And I'll also talk a little bit about how big data and the All of Us research program can address some of these deficiencies and disparities. In terms of the research we currently have available, this is the chart that essentially describes how many participants we have that are of different races and ethnicities. You'll see here that well over 80% of the population that we have genetic studies for are white. There's a number of reasons for this. That's a whole other talk, right? But you compare this to the global population, 12%, and so there's a massive disparity there. That's one that this is well recognized. This has been known for quite a while. And one of the issues is that, well, most of the genetic variability that we have within the world is outside of that one race group where we have 80% of the data. So what are we missing out on? That's one of the key questions here. And I'll present an example. PCSK9 inhibitors. So this is, PCSK9 is a gene that was discovered when researchers looked in African American individuals. The variants that led to this discovery do not occur in white individuals. So this is a concrete example of where, when we look outside of that race group, we make more discoveries. There's more genetic variability outside of the European ancestry group. So this is, it's not just about combating this disparity. It's about good science. It's about how this makes sense. If you're going to tie a genetic variant to some discovery, you might as well look in groups where there's lots of genetic variation, right? And so here's an example where this is something we wouldn't have found. This is a drug class that we wouldn't have that benefits everybody. It can be used in anybody, right? But the discovery that led to this was done in non-white individuals. And here's data to back that up. So even though we have a high proportion of individuals in genetic studies, the number of discoveries that are made, the number of associations that are made, are disproportionately in non-white individuals. So we have that data to back up the fact that we make more discoveries. We can find more classes of drugs, and we can find new associations when we include more diverse individuals and stop just looking in one race and ethnic group. So it's a distinct possibility that we may have largely exhausted all of the discoveries that we can make within a single ethnic group. So how can big data help? And what do we mean by big data? So there has been a recent explosion of data produced. We have multiple techniques where we can very cheaply sequence DNA. And DNA is a good example because we're talking about genetics here. So let's put this into perspective. The first human genome, which was finished in 2003, took about 10 years, utilized 20 sequencing centers, and cost almost $3 billion. Today, I can sequence your genome at one center for $500 in less than a day, right? So that's a big, big change. And clearly, it's creating an explosion of data. To put this into context, that's like paying not $70 million for a private jet, but $15. That's how much cheaper genomic sequencing has got in the past 20 years. So how do we leverage this data? How can big data help? Well, traditionally, researchers have restricted their investigations to a single disease. They investigate, say, high blood pressure, and they look specifically at that disease. What we've seen more and more is utilization of electronic medical records. So EMRs have data from every condition, virtually. Anything that you've been seen by your physician in this health system is contained within these EHRs, these electronic health records or electronic medical records. And so there's been a number of recent efforts to combine EMRs and genetic data. And because genetic data is static throughout your lifetime, you only have to do the genotyping once. And so here's a number of examples of consortia or initiatives where that EHR data are being tied to genomics. And that can lead to discovery. Now we have a virtual limitless clinical endpoints. We have lab tests. We have ICD-9 and 10 codes, which are essentially diagnostic codes. We have free text from different physicians. And then if we combine that with the genetics, then we can make those discoveries like this PCSK9 inhibitors. So narrowing down specifically to all of us, because the benefit of all of us is that it's diverse. And many of those other programs that I mentioned have other distinct limitations. The main problem is that they're not diverse. They contain mostly individuals of European ancestry. And All of Us was formed about 8 years ago with the specific intention to make this research more diverse. And as a corollary, to make more discoveries, to be able to identify new drug targets like PCSK9 and others. Just to note that we here at the University of Arizona are the leading national recruiter for this effort. So there are teams for this effort in California, in New York, in Florida, all over the country. This is a major NIH initiative, and we've recruited over 10% of the individuals for this initiative. So a lot of those diverse individuals come from right here in Arizona, Phoenix, Tucson area. And this is essentially how it works. There's an enrollment and a consent, and then EHR data is uploaded if consent is given, and then surveys are given to the participants, which are important. There are physical measurements that are done, samples that are provided, which are where we get the DNA sequencing from. And then there's also information that you can upload based on wearables and digital apps. Now, I want to call out specifically the survey data, because those other resources I mentioned, they don't combine EHR with genomics, with survey data, with patient-reported information. And this is important. Like, think of race as a self-identified feature. If you don't know what that person considers themselves, then you're inherently going to inaccurately define race or ethnicity. Social determinants of health, we're increasingly appreciating how these can influence health. Historically, when researchers saw, say, for instance, Black individuals have a higher prevalence of blood pressure, well, we would think, oh, well, it must be something in their genetics that makes them more prone to blood pressure. What we're finding out is that it's really more systemic inequities across our health system that contribute more to that disparity than it is to genetics. And so if you're going to find out the true cause, you need all that information. You need the survey data, the social determinants of health, the perceived racism, the socioeconomic status, and you need the genetics, and then you also need the EHR data. And so that's one distinct value that this program brings, is the ability to combine all those data sources. In terms of genetics, there are returns of results to participants. There's a number of different reports that participants get. One is a hereditary disease risk, so this is like rare variants, familial diseases that tend to be rare. In fact, you expect only about 2% of the people that you genotype, the people that are tested, to have a hereditary disease risk variant. However, I'm more interested in the medicine in your DNA. So I look at precision medicine, and so these are 7 genes that have impacts, known impacts, on drug response or side effects. And you would expect 96% of the individuals in this room, in this community, in this state, 96% of them are going to have some variant that changes their response to a drug or changes their likelihood of getting an adverse drug reaction. And here's an example of one of the reports, just to show you a concrete example. There's a lot of what we call star alleles and gene names here, but just to say that for this particular patient, there's a decreased function in a gene called SLCO1b1, and so the guidelines would recommend against certain statins in this participant. So the example that was shown earlier in the presentation where we had individuals that may or may not benefit or individuals that got side effects, those 3 blue individuals may have this variant and thus be steered away from those statins that carry a higher risk of those side effects. And that's really what we're all about. This is that getting away from that guess and check approach. Okay, let's give you this med. Oh, it didn't work. Okay, let's try something else. It didn't work. Let's try it. I mean, that's trial and error approach. That's not a very sophisticated way of curating drug therapy, right? So we want to use what data we can in a responsible way to determine who is going to respond to these different drugs. As a corollary, we're increasing efficacy, we're increasing safety for everyone, and not just a single race or ethnic group. But I'm going to leave you with this thought, right? So I mentioned data. There's so much data being produced here, and it keeps going up and up and up. And we're not just talking about electronic medical records and genomics. It's also what you ate that day, or the stresses in your life, or all of this multitude of factors that we're increasingly able to capture as we have the internet and we have security cameras and facial recognition software and all that stuff. It's not without its problems, certainly. But just think about our ability to process that data. It's not going up at all, right? And some would argue that it's actually going down. But this really creates a role for artificial intelligence and machine learning. And transitioning to Dr. Wheeler's talk, this is one of the forefronts, really, one of the places where we can be at the vanguard of precision medicine and healthcare is being able to process this data in a meaningful way. I want to thank the Kincoit College of Pharmacy, especially the participants for the All of Us research program, of which I am one. I want to thank the funders as well, and the great people in my laboratory. And with that, take any questions.
And I have a microphone here. So thank you, Dr. Karnes.
Thank you.
First question over here.
I have a couple of questions.
Okay.
So first, how does this program go into data protection and data privacy, as I believe most of us are concerned with,
Sure.
Just how our data is being protected?
So excellent question. So one of the focuses of all of us is on participant privacy. Obviously, with genomic data, all these self-reported outcomes, it's going to be a concern in terms of privacy. So one of the ways is a prohibition of stigmatizing research for instance, different race and ethnic groups or non-traditional gender individuals. Another way is a prohibition of re-identification. So you are not allowed to go in and use the data to re-identify individuals. The data is also protected by a certificate of confidentiality. So even if subpoenaed, the data is not dispersed to even a government entity, right? And so that's for NIH-funded studies. And so those are some concrete examples, but I've been impressed with the philosophies of the program in terms of their commitment to protecting data, because I think they understand that all it takes is a breach and then everything just falls apart, right? I mean, you ask many of the larger companies about that, right? So that is paramount to prevention, and so restricting access to responsible individuals is one of the priorities as well. And actually, it can be frustrating as a researcher too, because a lot of that data is usable. Like if I had 5-digit zip codes on everybody, then I could correlate, different states and different rural versus urban, but they don't allow us to do that. They only give us 3-digit zip codes in order to secure patient privacy. And so that does hinder research somewhat, but it's worth it if it means people are willing to participate.
And also, with big data as well, there's going to be a lot of information, of course. And so all that's processed and going to be used for the drug screening. And so what would you do with partial patient data if they don't consent to genomic testing or don't answer all the questions on the survey? What then?
Well, so, I think from a researcher standpoint, this depends on the analysis that you want to run, right? So certainly if you didn't get the genomic sequencing data, then likely that individual is going to be not included in any of the analyses that are targeting, sequencing data. Similarly with the EHR. And because this is big, right? So there's over 600,000 consented individuals here. So the proportions of individuals that you're talking about are substantial, but they're still allowed to participate, and they do. So there's small proportions of individuals that don't consent to EHR, that don't consent to genomics, and that's fine, and that's totally up to them, and they can still participate. So yeah, from an analytic standpoint, that will decrease the number of people, but again, they still allow individuals to participate.
So my question goes back to something you said about people of Black ancestry tending to have more high blood pressure, which I do recall from my schooling. But you said that you've discovered that has more to do with socioeconomic situations. So is that your data that found that? Is that something?
Specifically? No. So that's actually well established and predates the All of Us research program. So that's not a discovery that we had made. I was trying to highlight inelegantly that really we've assumed that anything associated with race has a biological reason for it. When in reality, what we're finding is when you account for those sociocultural reasons, they typically outweigh the biological. We have more similarities from a biological perspective, and the racial and ethnic inequities are largely driven by health disparities.
Okay, thank you.
Presentation. Thanks.
Thank you, Dr. Karnes.
Sure, thank you.
So our next topic is accelerating drug discovery with artificial intelligence. Drugs are used to treat, cure, or prevent diseases by binding to proteins in our cells, which involves specific shape, size, and surface features. Predicting how well a new drug can bind to these proteins is a complex challenge in developing new treatments. In this talk, Associate Professor Travis Wheeler will describe efforts to build computer software powered by modern methods in artificial intelligence that will accelerate the pace and reduce the cost of drug discovery. Please join me in welcoming Dr. Wheeler.
Tap, tap, is this thing on? Yes. Thanks for that nice introduction, and thanks to all of you for coming here. I've been wandering around this building for a couple of years thinking, what an amazingly odd place to give a talk. Especially that place, I guess. But now I get a chance to do it. I'm pretty excited to talk with you about this particular topic. So this is a lecture series that's put on by the College of Pharmacy. So I assume you're here for the drugs. Maybe not. Maybe you're here for the artificial intelligence. And if you're here for that, you've all benefited in some way and maybe been annoyed in other ways by things that are tied to artificial intelligence, and you probably mostly don't really know what it does. It means to you about as much as this picture does to me, which is that it's something fancy. So let's talk a little bit about this whole topic of drug discovery and artificial intelligence, we're going to talk about it in 3 parts. So first, I'll talk with you, give you kind of the quickest primer in the history of the universe on what is artificial intelligence, and then a little bit of a primer on what is drug discovery, what do we mean by identifying drugs, and how do they interact with the proteins that they tend to interact with. Then we'll kind of tie it all together and try to understand a little bit about how artificial intelligence influences drug discovery. I'm not going to talk much about that word accelerating after all. So what's artificial intelligence? I'm going to read from a slide, right? At AI, it's a broad field of computer science that aims to create machines or computing platforms that can perform tasks that would typically require human intelligence. So that could be recognizing patterns, automating control of physical systems based on those recognitions, like self-driving cars, understanding language, maybe responding to that language, like those really helpful phone systems that you interact with when you call the hospital and you want to talk to somebody who's a human. Creating pictures or music. This is the new wave of amazing stuff between conversations that you can have with a computer on ChatGPT and the ability to create pictures. And that's a really broad field, and we're going to focus in on one particular subfield of AI called machine learning, which is designed to effectively teach computers to learn from data, right? So to have a machine learn something. And I'm going to give you a simple example of this first, which is actually a really easy thing to do, which is to decide if a picture that I give you is an apple. You're all amazing, because you can all look at that picture and you know it's an apple. Or at least it's a picture of an apple, one of those, okay? How would you tell a computer to decide if a picture is an apple? You get a picture. You might say, oh, it's round and roundish. It's got a stem, and maybe it's red, and that's a pretty good description of an apple. That's even a hard problem, just to do what I just described, to define how do you describe that something is round, or how do you describe that it's roundish, and what's a stem? Those are hard things to do, but they're not even good enough, because there's an apple. That's red-ish, right? There's an apple. That's definitely not reddish. There are apples there, but it's not an apple, right? If I've got a program that's supposed to guess that that's an apple from the description that I just gave you, it's in trouble. Right? Is that an apple? It's on my computer. Okay, is that an apple? No. Also not an apple, but it really meets the round and red and has a stem. So the problem is, right, you've got to somehow be able to teach a system to be able to understand how to classify a thing. In this case, it's how to classify something as being an apple. And there's no way on earth you're going to effectively be able to do this by explicitly programming the computer to perform that task. And so what we do instead is we develop some kind of a model that is able to learn how to perform a particular task, in this case classify a picture as an apple or not an apple, by showing it lots of examples of the data and then letting the system figure out what patterns it needs to be able to observe in order to be able to effectively identify those. Now I'm going to actually simplify this problem. It looks a little harder, but actually it's a lot easier. We're going to look at a bunch of handwritten digits that are just in black and white, and they're just a few pixels for each one. These are 0s and 1s and 2s and 3s. And you can all identify almost all of these perfectly if I just showed them to you, right? But it's actually not that easy a problem, and it took years for people to develop methods to be able to do it effectively. And the problem boils down to something like this. I have a hand-drawn picture, and it's pixelated, and I can look at the brightnesses of individual pixels, and the brightnesses can somehow, by the computer, be used to recognize particular facets of the picture, like, ooh, here's a little edge that's sort of like got this kind of a diagonal shape to it, and to be able to piece those together in some way so that it can recognize what a particular digit is. Like, for example, a circle. Maybe you could imagine that a system might think of it as being composed of these individual little pieces, and if you could just somehow have a system that would recognize that all of those pieces are present and can compose them into a single circle, then you'd know you've got a circle. And maybe you also have some other thing that can recognize when you've got vertical lines, and you can compose those. I've got a circle and a vertical line, that's probably a 9, and I've got a couple of vertical lines and a horizontal line, that's a 4, and so forth. Oops. And this can somehow, we hope, maybe be able to piece together what the digits are. Okay, but how do we even do that? How do we make the smaller things that compose into more complicated things that compose into more complicated things? And the lessons that that the community has devised for doing this are stolen or taken from the way our nerves communicate information from one place in the body to another place in the body via these neural networks, right? These neurons that capture some information and then pass it on to another neuron that composes information from multiple neurons and passes those on. The visual system works in the same way. Your eyes are doing the same thing as we're talking about here effectively. And we developed these things that in the machine learning community are called artificial neural networks that are really intended to sort of replicate the core idea of how neurons work. And you can imagine in this case, for example, that we can have a layer at the very beginning that is, what are the brightnesses of all the pixels in the picture? And then another layer that says, okay, well, I can see the top neuron there. It's a neuron that recognizes particular signals. I can see a little horizontal thing that's sort of like the pink thing on the top. And I've got another neuron, the third one down, that's blue, and it's good at recognizing this thing that's mostly vertical, but a little bit of an angle. And each of these neurons recognizes particular kind of angles or rotations of structure. And the next layer over to the right has neurons that recognize larger composed elements like circles and vertical lines. And then the next one composes those to form numbers. So I've sort of lit up the particular fields that correspond to recognizing a 9 from just a bunch of pixels. And you might imagine that that's actually what the neurons in that neural network that I just showed you are figuring out. It turns out they're not. They're actually seeing something more like this nonsense that's in the lower left corner here with all kinds of patterns that are definitely not how I would imagine that a computer would figure out what the various digits in the handwritten alphabet are, but this is what they figure out, and there you can use it to very effectively match the human annotation of what a 1 and a 2 and a 3 are. I'm going to stop talking about that particular method, and I'll just point you to this great website, great YouTube series, 3 Blue, 1 Brown, if you find yourself thinking, I really want to know how all that works. Okay. So that was AI a little bit, super fast, right? Basically, exposed to data, learn how to recognize patterns from those data. Drugs. Okay, what drug is that? What drug is that? It's ibuprofen. I had a headache earlier today. I took one of those. Okay, so this is ibuprofen. Okay, and it's just a molecule. It's got some carbons and hydrogens and a couple nitrogens in it. And it's not actually just a bunch of lines drawn on a page. It's a three-dimensional structure of sorts. And one way we can look at this particular drug is to look at it in this ball and stick formation that shows where the carbons are and the rotational angles that occur. But even this doesn't really represent what the molecule looks like, because it's got this electrical field, or the electron field around the various atoms. And in some ways you can think of its interaction potential with the world around it in this kind of a weird caterpillar kind of a form. So that's going to be a representation you're going to see for a few more slides down here, is this is a drug that is able to interact with the world around it. Okay. Molecules can be a lot bigger than the little tiny ibuprofen that I just showed you. This is an albumin protein. That's the predominant protein floating around in your blood. And you can see it's a lot more complicated than that very simple thing. Sitting inside of this one little binding pocket is an ibuprofen molecule that has gotten stuck in there. And you can see it sort of looks like a puppy if you look at it in just the right angle. And it sits inside of this little cleft in the protein that's called a binding pocket. And that is where drug interactions occur. It's one of the places that drug interactions occur in this particular protein. And if we want to drug a protein, what we're looking to do is to say, this is another representation, it's a ribbon representation of the same protein. If we want to identify drugs or understand how drugs are interacting with the proteins, we're looking to recognize these pockets in proteins, binding pockets, and guess or understand or predict where particular drugs might fit, which ones might fit into those pockets. Okay, so that's this. This is the task of drug discovery. One way to do it is to just throw a million chemicals at a protein and see which ones stick. That's an exceptionally expensive thing to do, but it's what all the big drug companies do if they want to make a drug that will sell for billions. Another way to try to do it so that we can make drug search faster and more expansive is to use computers to try to predict for a given target protein which of the, at this point, tens of billions of drugs that we can conceive of, don't actually exist, but we can conceive of making, which of those are likely to bind into that particular pocket and have some kind of an effective docking outcome like what I'm showing here. I want to focus on one more particular protein just to give you another example of what it looks like, because I think this is a pretty important example. What I'm showing here is a really simple schematic of a thing called a G protein coupled receptor, a GPCR. It lies at the membrane between that encompasses a cell. Right? Everything that's outside of the membrane is extracellular. Everything that's inside of the membrane is what makes your cell do what it does. You don't want to just puncture a hole. If you want to recognize that something is out there, you don't want to puncture a hole because then the insides bleed out of the cell. And so you have these membrane proteins that have a receptor that receive some particular molecule, a drug in many cases. And in response to receiving that particular drug, they change the conformation a little bit on the inside, and it causes something new to happen inside the cell. Okay, in this particular case, the drug or the ligand binds that little red ball on top, and it causes the G protein, which is the thing that's inside, to change in conformation a little bit, and it lets go of its ADP and picks up an ATP, and it dissociates, and then the resulting components go off and do their thing, which can be different depending on which particular drugs we're talking about, or which particular GPCRs we're talking about. This is a slightly prettier picture of the same thing. It's not actually a linear thing like this. It actually kind of forms a basket, and you can see the stimulus is the pink thing on the outside, and it's made something happen on the inside. And here's an even prettier thing that's not even a picture anymore. It's a video that is just a video representation. No, this is not a real recording of a thing, but that light green thing just bound to the GPCR that's dark green, and the GPCR now has changed in its conformation, and it can bind to the G protein, and poof, there goes one of the ADPs, and then it's dissociated, and the red thing goes along, and it does its fancy thing. And some magic happens here, and it doesn't even matter what the magic is. Something happened because a drug was received on the outside of the cell, and now things are happening on the inside of the cell. GPCRs, there are about 1,000 different GPCR proteins in your genome, which is like 5% of your genes, and they're the target of roughly 40% of FDA-approved drugs on the market today, so they're a pretty big deal. Right? They're not the only kind of proteins out there, but they're a very interesting collection of proteins, and what you just got to see is that they both do bind to things, but also that they actually wiggle and move, and that's a really important thing to remember. These are dynamic molecules, they're not bricks, and that affects drug activity and also drug discovery. So now I want to put these things together again. So what is machine learning? How does it apply to drug discovery? So the task, remember before we had the task of identify an apple? Well, now the task is I'm given a protein and I'm given a drug candidate, predict whether the drug is going to bind. It's going to go in and stay attached to that particular location. That's a lot harder than is it an apple, right? I hope you believe me. Somewhere here. Okay, so how are we going to train this model? Before we said, let me train it by showing it a bunch of apples. Here, there's the video. We're going to train it by taking a large database. This is what people do right now, what I've done when I've built machine learning models to try to predict drug interaction potential, is to take a ton of known protein structures, of the complexes between the drugs and their target proteins, and learn something about the way that the drugs interact with their proteins. Okay, here's a heat shock protein. It's a particular protein that we've got a few dozen known drugs that bind into this particular pocket, and each one fits in just the right way to that particular pocket, and it's got interactions. There's hydrogen donation or the hydrogen electron donation to the other atom, or it accepts it, or there's a pi stack interaction. There's a variety of chemical relationships between these two, between the drug that is set in there and the protein that it's bound into. And they happen in a particular location relative to that particular protein. And it turns out that particular locations are fairly well classified. So you can see there are these regions that are blue, which is one of those interactions, and regions that are orange, which is another interaction. These are the places that particular kind of interactions occur. And we can learn where those interactions occur, and given some new protein, we can try to predict where are the same kinds of interactions likely to occur. And I tend to think of this as sort of like, if I've got a pocket, I'll put a bunch of little magnets throughout. And the magnets will be oriented this way or that way. And if I put a drug in here and it's got little magnets oriented in this way and that way, and if I got the wrong orientation, it'll go in and get pushed right out again. If you've ever tried to take two magnets and put them the wrong way, they push away from each other. But if I get them just right, it'll actually go in and stick. These are more complicated than magnets, but it's the same idea. Do I get the correct collection of forces together in the correct location? Most drugs, the answer will be no. A few, they'll be yes. And can we predict them based on this classification? The way we get that is by showing our models a bunch of proteins which have been exposed, for which we have a few known drugs that actually bind, maybe a few dozen if we're lucky, and thousands or tens of thousands of drugs that don't bind. And so we learn in the same way we would say, that's an apple, right, by showing hundreds of pictures of apples, and this is a not apple by showing millions of pictures of things that aren't apples. We can have a model learn whether it is or isn't going to bind. That's the hope. It should work. I've convinced you machine learning is great. You've all seen self-driving cars, right? So those are super cool, right? This is a relatively new thing that you can do with computers. You can say, or you can type, create a scene from a mountainous country village. We're watching a group of hot air balloons made out of roses, and computer made that picture, right? No person made that picture. A computer made that picture from a verbal prompt like that, which is miraculous, okay? I really like this example. I personally wanted to create a couple of haiku poems to present my work. And so I asked it, I said, hey, ChatGPT, this fancy new thing, I run a research group, and my research is described on this page, gave it my website. I'm giving a talk today about our work in drug discovery. Please create a pair of haiku poems that combine together, right, to describe my research. And I put a question mark there for some reason that I don't know. Okay, and it came back, nature's mysteries, decoded with computational methods we create. From genomes to drugs, from microbes to behavior, our work never stops. It's actually a really good description of my work from a kind of a complicated text. This is amazing, right? A computer did this just by looking at my website. It's really cool. It's wrong, because it's not a haiku. It's got nine syllables in one of those things, and now I'm getting really picky because it's really amazing. The other thing to know is I tried it 20 times before I got one that was halfway decent. The first 19 were terrible, but it's still 1 out of 20 ain't bad because it made a haiku and it's just a computer. Here's a picture, a collection of pictures that I got back when I asked one of these photo creation things to create a photorealistic image of a person holding a hammer and cutting a thin rope with it. There's reasons for that I'm not going to get into, that I really wanted that picture. It did not give me a picture of someone with a hammer cutting string. I hope you can see that they don't even have hands, right? Self-driving cars, if you've been paying attention to the news, in San Francisco, there was a recent case of one of these cruise self-driving cars driving into wet concrete. That's not great either. And this is really important because in San Francisco, most of the concrete is dry. So it's pretty safe to drive around, and only occasionally do you run into wet concrete. In the context of drug discovery, most of the drugs that you might be trying to place into a drug binding pocket are not going to fit. I'll think of those as wet concrete. The vast majority of your landscape is wet concrete. Odds are good that your self-driving car that's trying to predict if a drug is going to bind into a pocket is going to run into wet concrete and it's going to get it wrong. Okay, so it's a much more challenging problem, I believe, to discover drugs than it is to drive a car around in San Francisco. And there's a pretty large growing body of response to all of the hype about using machine learning methods to find drugs, which I've been a part of. And the response that recognizes that actually there's a lot of failures in this. And for the most part, you can't really rely on them yet to be able to solve the problem of drug discovery. But I think there's still reason for hope. And this is the last thing I'm going to talk about before I end this. Here's an example, actually, the pictures that I showed you before where they couldn't even make a hammer. And I've been trying for a year to get a picture of a hammer with a string, and I go back every couple weeks just because I'm interested in the state of the art. Turns out that this morning, for the first time in more than a year, I was able to get pictures that had hammers and string in them. It's still not what I asked for, but I'm getting closer. Right? So the models are learning because they're being exposed to a greater diversity of these kinds of problems. And the fundamentally what's happening is they're getting better training data. Right? They're getting exposed to more apples in more configurations. And in the context of drug discovery, one of the things they're doing is that we are finally recognizing as a community that you need to account for not just the diversity of what kinds of proteins there are, but for the fact that the proteins move, right? That they're dynamic. They wobble and wiggle, and they interact both just by themselves with their own atoms, and maybe more importantly, they interact and respond to the drugs that bind into them, right? So you can see in this case on the right that this drug is actually changing the conformation of the protein a little bit that it's bound into. And if you don't account for that, you're doomed in the realm of drug discovery. So all that's left to do, right, is to have a large database of these complicated, dynamic instances of these molecules, and to train the machine learning models to use them. Problem is, the database doesn't exist yet, but we're actively building it right now. I'm expecting to release the first version of this thing probably by end of year, give or take, and that's going to be a thing called Molecular Dynamics Repository. It's a repository of these dynamic molecules. It'll store millions of these things eventually. It'll take a while for the full community to contribute into it. And all of them are going to be available freely to researchers who can train their models or explore the dynamics of molecules to their heart's content with the assistance of a lot of the different sorts of things that we have available to us here at the University of Arizona between the Bio5 Institute, which is a building right across the way there, and Cyverse, which is the largest National Science Foundation funded computational infrastructure in the country, which is also housed here. All of this has been made possible, and I think there's cause for hope. Call it 5 or 10 years, and we should be able to use computers to do the thing that I just said we ought to be able to do. And with that, I'll say thanks. I think I probably ran a little bit over time. Apologies. And would love to answer any questions.
Any questions? Thank you, Dr. Wheeler. Oh, question right here.
So in the current state of AI drug discovery, what is the most common way for drug candidates to be generated? Is it like based off of like the current known functions of the proteins, or based on the amino acids in the binding pocket? Is it random? How is it generated?
When you say drug candidates, so it depends on how. So there are drug candidates in one way, which is there are a few databases of drugs that pharmaceutical companies say they could probably synthesize or create relatively cheaply and easily. And that there are databases of something on the order of 10 billion of those probably synthesizable drugs, most of which have not ever been created, which are in some way the candidates. Right? So those are not actually going to be tested for the most part, but they can be used by computers to predict drugs, right? Which of the subset of drugs is likely to be an effective binder. And then the next step would be to take this very seriously downsampled set of maybe a few hundred or a few thousand, have them synthesized, pay the money to do it, and then to perform further tests for binding. So I'm not sure if you're asking about the first question, which is just drug companies say they can make it, or the second, which is what are the methods that are used to identify those?
How does a computer choose which molecule to run in a simulation of binding with the protein?
There are 500 ways. I'm not going to list them all for you right now. The answer is basically there are a lot of ways that at the very most generic form use the method that I just described, which is they in some way recognize what the binding interaction potential is of a particular region, or what the likely forces are that are imposed if you pose a particular drug into a particular pocket, and to predict, usually pretty poorly, how well that drug is likely to bind into that pocket, right? But the specific mechanisms are every kind of machine learning model that you can imagine building either has been tried or will be tried at some point in the near future. That's a terrible non-answer, but mostly it's a lot of ways.
Yeah. Thank you for the nice presentation. One of the challenges of drug discovery and drug development is you go in there, you find an active molecule, but then you run into what the previous talk touched upon is unwanted reactions. Where do you see artificial intelligence going, helping to weed out these molecules that have unwanted reactions?
Yeah, this is an incredibly important question, right? So great, I found a drug that will bind into a particular pocket. I started with this video of ibuprofen stuck into a binding pocket in albumin. That's not the thing that you want it to bind into. That's an off-target effect, which is what you're effectively referring to, where it also happens to land in the albumin in addition to its other targets. And it causes a side effect, which is, among other things, accumulation of bilirubin, right, in the plasma. And that's an undesirable outcome. So the answer is, in part, right now, there's no hope of really being able to solve those. So you end up always having to get down to the point of having, at some point, clinical trials or preclinical trials involving animals to date. Long term, right, this is an area that I'm quite interested in. One of the things you would like to be able to do is to say, hey, I've got this drug. I think I found it being a good potential target for this particular protein, which is my target protein. Why don't I check it against every other protein that is constructed in the human body, right, and see if it's got any risk of binding to any of those as well, so that at least I've got a sense of what the potential risk is, right? Which is not a perfect answer to the question, because the complications of multiple protein complexes still make it so you can't quite answer all of the questions, but it gets you along the way. But it's an incredibly important issue that's not going to be solved in the next 10 years, I'm sure of that.
We're going to take one last question.
Such a cool talk. Question is that it's so amazing to be able to like use these models to identify patterns. It seems like essentially is what's going on. But how easy or how difficult is it to like look back and then have us understand how the model is recognizing the pattern, so then maybe we have a better understanding of the underlying biology or physics or chemistry that's going on?
Terrific question, right? So this is a question of the interpretability of models. You saw the picture that I showed of the digit recognition and the way these machine learning layers recognize really bizarre kind of patterns that don't make sense to me as a human. So there are a couple of issues at play. It's a huge area of research in machine learning in general, is both how to gain insight into what models are doing, so like peer back into particular components of the model and interpret what they have learned, but also to design models that are by design more interpretable, right? So I don't have any answers to the question of how we're going to do this in the context of drug discovery right now, but it's an area of research that is, I think it's probably safe to say that more people are working on interpretability of machine learning models in general than are working in drug discovery with computers in particular. But it's going to be, for the next 30 years, is my guess, an open topic for heavy research among the computer science community.
All right. Thank you, Dr. Wheeler. And thank you all again for coming for our first talk in the series. We hope to see you next Tuesday for our second series of talks that highlight pharmacy innovations in patient-centered care and the drugs associated with adverse outcomes when used long-term as we age. I encourage everybody to enjoy the dessert reception and see you next time.